really looking for those LC50, LD50 values, and those are the concentrations at which 50% of your population is going to die. But real-world exposure is chronic and it's low level, and these sublethal effects are not being studied and they're not being published. What we instead think is going on is hormesis, and this is now being almost considered a fundamental concept in biology because it is universal across species, across contaminants, across cell lines versus individuals, and it is a biphasic dose response where at high concentrations there's inhibition, there's toxicity, there's lethality, and then at low concentrations there's that stimulation of the adaptive stress response pathway and then also stimulated development as a result of that. So my research questions are, do forever chemicals alter when mosquitoes develop? And are measurable changes being detected in RNA sequence reads? So my study species are Culex pipiens. They are a temperate species of mosquitoes found on the eastern shore of Maryland. And I raised these in 3 different treatments. One was a mix of 17 different forever chemical congeners. And one was a 8 carbon chain, long chain, and then one was a C4 short chain. I raised them in concentrations. I really wanted to get the full dose response in my study. So concentrations of 0, my control, 10, 500, 1500, 3000, and 5000 nanograms per liter. And then for my individual doses, just 100 instead of 10. And then I also fed adults, regular adults, blood that was spiked with these forever chemicals at concentrations of 0, 10, and 1000 nanograms per liter. I tracked these developmental parameters every single day throughout the course of the study, which was on average 32 days. I kept track of pupation, adult emergence, and then feeding behavior. The statistical analyses that I performed were Cox proportional hazard models, Kaplan-Meier survivor analyses, negative binomial generalized linear models, and RNA-seq processing using HiStat2 and GFF-read. So a hazard model is a method to analyze the time a specific event occurs. And from the recommendations of ecotoxicology is not normal, generalized linear models and hazard models are a better fit for ecotox data as opposed to non-parametric tests because they explicitly account for a non-normal error in structures and mean variance relationships. So diving into my results, first here I have a hazard plot of my mixed treatment groups. So how quickly that they reached pupation and how quickly they reached emergence. So there's not a ton of visual differences in my pupation from days 0 to 20, but I really want you guys to focus on is all of the differences that we're seeing in adult emergence. Okay, specifically days 13 and 14, which are highlighted by this band here, every concentration group relative to control is significantly stimulated. There are other days which other concentration groups are significant relative to control, but on these 2 days, every concentration group is. But hormesis doesn't just stimulate growth, it also redistributes growth timing. So we wanted to create 2 windows through which that we could investigate timing and development. So we had an early window, which was days 5 to 15, and a late window of days 16 to 30. So this is an odds ratio plot of development during these windows. Anything shaded to the right of the dashed line is stimulated. Anything to the left of the dashed line is inhibited. Here, what you can see is for emergence, as you increase concentration group, we're becoming more stimulated up until 5,000 ppt, where it's no longer statistically significant. When I want you guys to think about the hormesis curve, the dose response is as concentration increases, adaptive response is increasing as well until it becomes overwhelmed, and then you have that inhibition. So more evidence for hormesis. We also want to investigate this through the median time to developmental days. So back into our pooled data, what you can see here is for that both pupation and emergence, our control has the highest median day to both of these developmental endpoints. As you increase in concentration, that median day drops and drops and drops until you reach 3,000 ppt, and then it rebounds up at 5,000 ppt. Once again, that U-shaped curve that is indicative of hormesis. We wanted to see if these larval responses were also going to be translated into adult behavior. So next, when we fed our adults the same 17 forever chemical mixture, what we saw was at 10 ppt, which was, supposed to be our proxy for domesticated animals, our mosquitoes fed 89% higher relative to control. At 10 parts per billion, which is supposed to be the levels that are what we see in humans and our more sensitive wildlife, they fed at 91% higher rate relative to control. And both of these values were statistically significant. We also wanted to see